Hello, everyone, and welcome to today's EBD Group Bioeconomy webcast titled Maximizing Human Predictability from In Vivo Pharmacology Studies, sponsored by Axelid Drug Discovery Partners. This is part 3 of our 3-part series titled Transform Your Drug Discovery Faster Paths to Clinical Success. First, I'll cover some quick housekeeping items. If you experience difficulties with audio or advancing slides, refresh your screen with F5. If you are experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, you can submit your questions into the Q&A window on the left-hand side of your screen. And lastly, in 24 hours, you'll receive a link to watch the recording of this session. Let's now begin by introducing our very first speaker for this session. We have Manami Kaneko, PhD. She's a Senior Director of Pharmacology Business Unit for Axelid Drug Discovery Partners Incorporated. Thank you for joining us today. I'll now hand it over to Manami to begin the session.
Okay, hello everyone, and thank you for joining today's webinar titled Maximizing Human Predictability from In Vivo Pharmacology Studies. I'm Marami Kaneko, Senior Director of the Pharmacology Business Unit. Together with my colleague, Dr. Yasunori Neo, we'll share insights into how Axelid enhances translational relevance in preclinical studies. Before going into the main topic, please allow me to briefly introduce our company. Axelid is a Japanese integrated drug discovery solution provider. We are a unique drug discovery CRO, founded as a spin-off from two pharmaceutical companies, Takeda and Teijin. Our team includes more than 300 scientists with deep pharmaceutical experience. We also maintain a compound library of over 1.2 million compounds. One of our key strengths is access to over 1,000 drug discovery project database inherited from Takeda and Teijin. These valuable resources support our clients' research by enabling data-driven decision-making and efficient development strategies. Axelid offers a comprehensive drug discovery platform covering everything from target identification to IND and NDA-enabling studies. We combine legacy data and generative AI technologies. This integration boosts innovation and success rates. Our capabilities also span modality expansion, and we continue to strengthen our network through strategic partnerships. Let me introduce the key features of Axelid's pharmacology platform. We support research across more than 7 disease areas, including pain, immunology, cancer, CNS, cardiovascular, metabolic, and bone and joint disorders. Our platform is designed to evaluate a wide range of modalities, extending beyond small molecules. We operate a fully RX-certified animal facility. This facility enables studies using mice, rats, dogs, pigs, and monkeys. At Axelid, we provide a broad selection of disease models and evaluation systems, including both standard and customized approaches tailored to specific research needs. Importantly, we have an extensive track record in conducting experiments for IND application. We include specialized teams in gene-modified animal creation, omics and bioinformatics analysis, and pathology. These capabilities enable us to support your research from POC studies to translational research. Bridging the gap between preclinical and clinical development. Today, I will begin by presenting an overview. Following that, Dr. Yasunori Nio will share case studies in cardiovascular and metabolic disease. Let's begin today's main topic. As you know, results of animal models often fail to accurately predict human drug efficacy. According to a 10-year follow-up study of drugs that showed efficacy in animal studies, as shown here, only 5% were ultimately approved. And these numbers apply only to drugs with published animal data, so the actual success rate may be even lower. We believe there are 4 key factors contributing to this low translational success. First, poor reflection of human pathophysiology in disease models. Second, model selection based on convention rather than data. Third, inconsistent evaluation parameters between preclinical and clinical studies. Fourth, incomplete understanding of safety and a mechanism of action. To address the challenges, Axelrod has developed several key strategies to enhance the translational relevance of animal studies. These key strategies are customized disease models, clinically translatable models and assays, clinical diagnosis tools and parameters, and comprehensive evaluation, efficacy, safety, and MOA. I will introduce our strategy, which begin with these 4 C's in order. Let's start from customized disease models. As you know, there are significant genetic and physiological differences between rodents and humans, which often limit the relevance of traditional animal models. As Axelrod, we address these challenges through advanced model development, including humanized mouse models. These include mice with human immune system, human hepatocyte chimeric mice, and human gene knock-in mice. We use pig and non-human primates, which offer a closer physiological similarity to humans. Another challenge with conventional models, like spontaneous disease development, surgical indications, or chemical dietary manipulation, is that they often suffer from low reproducibility and limited genetic specificity. To address this issue, we propose the creation and utilization of genetically engineered rodents. Let me introduce Axelrod's humanized immune system mice, which are powerful tools for evaluating immune-targeted therapies. We use various types of NOG mice, which are highly immunodeficient due to IL-2 receptor knockout. By engrafting these mice with human PBMC, or human hematopoietic stem cells, they develop human immune cells. Axelrod has developed a unique enhancement method using hydrodynamic gene delivery system. By introducing human IL-15, which is essential for NK cell maintenance, we have successfully improved NK cell engraftment like this in these models. We offer various models with different immune cell profiles to support translational assessment of drug efficacy and safety in humanized immune environments. Next, I'd like to introduce an example of human gene knock-in mouse, the human transferrin receptor knock-in mouse. The transferrin receptor is expressed on vascular endothelial cells of the blood-brain barrier. When its ligand binds, the receptor is internalized via endocytosis, and the ligand is subsequently released. In this knock-in mouse, we confirm the expression of human transferrin receptor in brain vascular endothelial cells, like this. This mouse model is highly useful for screening human transferrin receptor antibodies or peptide tags designed to cross the blood-brain barrier. In this way, Axlead creates human gene knock-in mice to support projects targeting species-specific proteins, enabling more accurate and translationally relevant evaluations. As the final example of our customized disease models, I'd like to introduce our large animal models. Axlead has developed and utilized immunosuppressed pigs and monkeys to evaluate cell therapies. Immunosuppression was achieved by removing immune organs and giving immunosuppressive drugs. Under these conditions, we successfully induced type 1 diabetes or heart failure, followed by cell transplantation. We confirmed that the transplanted cells were engrafted and functionally active. We use large animal models to provide more clinically relevant evaluations. One of the key challenges in drug discovery is selecting appropriate disease models and assays that truly reflect clinical conditions. To address this, Axlead leverages its extensive experience and deep expertise gained from over 1,000 drug discovery projects inherited from Takeda and Teijin. These projects span a wide range of therapeutic areas and target classes from early stage to lead optimization. By combining real-world data with scientific insights, we help our clients make informed decisions and improve the success rate of their drug development programs. To further improve model evaluation, Axlead has introduced AI-based behavior analysis to optimize parameters and detect subtle changes that were difficult to observe visually. For example, in a schizophrenia model, a conventional visual observation allows us to assess only negative symptoms, such as reduced sniffing behavior. By applying AI analysis, we were able to detect changes in grooming frequency and duration, indicating the presence of positive symptoms like schizophrenia patients. AI-based analysis enhances the sensitivity of behavior studies, supporting more accurate and translationally relevant assessment. Next slide shows, as you know, differences in diagnostic tools and biomarkers and evaluation criteria often lead to discrepancies in data interpretation and hinder translational success. To address this, Accelerate applies clinical grade diagnostic tools like here and parameters in animal studies. This approach improves data consistency and enhances the translational relevance of preclinical findings. The details will be introduced in the next section by Dr. Neo. Despite promising results in animal studies, some drug candidates with unknown MOA fail in clinical trials due to insufficient efficacy and unexpected side effects, such as MET-AP2 inhibitors. And some drugs also caused potential side effects due to targets in the clinical study. A good example is GLP-1 agonists, which have become first-line treatments for obesity. The detail of GLP-1 agonist will be introduced in the next section by Dr. Neo. To improve clinical success, it is essential to conduct comprehensive evaluation in preclinical studies, not only efficacy, but also analyzing MOA and potential side effects. In summary, Axelis in vivo pharmacology platform is designed to maximize human predictability in drug discovery. By integrating customized disease models, clinical diagnosis tools and markers, and comprehensive evaluations, we help our clients improve the success rate in the clinical setting. Thank you very much for your attention. That's all my presentation.
Thank you, Manami. Let's take some questions before we move on to Dr. Neal. Our first question, Dr. Kaneko. Can you create knock-in mice with long DNA inserts, say over 10 kbp?
Thank you. Interesting question. Yes, we can. Inserting long DNA sequences over 10 kilobase pair is technically challenging, but it's definitely achievable with our appropriate strategy. We've successfully completed several projects involving large knock-ins, not only in mice, but also in rats. In some cases, we've inserted human gene sequences while preserving the original exon-intron structure, which is especially valuable for evaluating nucleic acid-based therapies. Thank you.
Thank you very much, Manami. Another question here. What other assessments can AI analysis perform?
Thank you for your interesting question. We are currently developing our AI analysis capabilities in collaboration with our partners. In addition to the example I mentioned at this seminar, our system can also detect behaviors such as itching, pain-related responses, and seizures. We are actively working to improve the accuracy of these analyses so they can be reliable used in the real-world research. Thank you.
Thank you. And our final question before we move into Dr. Yasunori Neo. Manami, can you please
Yes.
share case studies in which you measured the same devices and markers as in clinical settings and obtained results comparable to those in humans?
Great question. Thank you. One example in the evaluation of cardiotoxicity caused by anti-cancer drugs, which is a major concern in clinical practice. In hospitals, echocardiography is commonly used to assess cardiac function. We applied the same method in rodents treated with doxorubicin, a widely used chemotherapy drug. Interestingly, we observed that global longitudinal strain, GLS, decreased earlier than ejection fraction, just like in human patients. This kind of result showed using clinical grade device and markers in preclinical models can provide highly translational insights. I think so. Thank you.
Thank you very much, Manami, for an excellent segment. And let's bring in our second presenter today, Yasunori Neo, PhD. He's the Director of Pharmacology Business Unit for Axley Drug Discovery Partners Incorporated. Welcome, Yasunori. Please begin.
Yeah. Hello everyone. My name is Yasunori Nio from Axelid Discovery Partners. Today I will present case studies in the cardiovascular and metabolic disease area. The goal of my talk is to illustrate Axelid's unique strategies to maximize human predictability from in vivo pharmacology studies. I will explain customized disease models. In obesity, showing blue, human diseases is largely driven by multiple environmental and lifestyle factors, making conventional diet-induced obesity DL mouse is a reasonable approximation. However, our MC4R knockout mouse provides distinct advantages. Although genetically defined, it enables rapid evaluation of anti-obesity drugs and importantly respond to clinically established GLP-1 agonist. This strong pharmacological concordance highlights its translational relevance. In MASH, showing green, human pathology arises from mainly Western lifestyles. The conventional CDAA high-fat diet model induces steatohepatitis but doesn't capture MASH multifunctional factorial etiology. Our Western diet MC4 knockout mouse combines genetic sensitivity with dietary triggers, closely reproducing human metabolic syndrome and progressive liver disease. In addition, this mouse model was highlighted as one of the top 5 most translatable MASH model in the Nature Metabolism journal. In chronic kidney disease, CKD, shown red, conventional models such as nephrectomy can mimic renal failure but lacks the natural cause of disease progression. Our collagen 4A5 mutant mouse develops proteinuria, fibrosis, and renal functional decline, features broadly shared with human CKD. In summary, while conventional models capture limited aspects of disease, our portfolio, MC4 knockout, Western diet, MC4 knockout, and collagen 4A5 mutant mouse provides rapid and pharmacological translatable models across obesity, MASH, and CKD. These platforms offer superior value for clinical drug discovery. This slide shows that I will explain clinically translatable models and assays. First, let me introduce our obesity model using MC4 knockout mouse. These mouse developed severe obesity, shown here, and insulin resistance, closely resembling human obesity. We tested 3 GLP-1 analogs, including semaglutide, tirzepatide, and retatrutide. All GLP-1 analogs showed significant reduction of body weight and food intake. Tirzepatide was the most effective, followed by retatrutide and semaglutide. These results were along with clinical efficacy dependent manner. Similar with clinical study, GLP-1 analogs decreased both fat mass and lean mass. And according to X-ray CT analysis, visceral fat, shown pink, was greater, reduction of the visceral fat was greater than that of subcutaneous mass, shown yellow, by tirzepatide treatment. By Oxymax system analysis, GLP-1 analogs shows reduction of the energy expenditure, primarily due to the effect of appetite suppression. Our second case is focus on the MASH. Western diet-fed MC4 knockout mouse develop progressive liver disease, moving from steatosis to fibrosis. Closely reflecting human MASH pathology, we examine the effect of clinically approved drugs, resmetirom and semaglutide, and using this mouse model. Both resmetirom and semaglutide improved steatosis and fibrosis, while only semaglutide reduced energy expenditure due to food intake suppression. On the other hand, resmetirom improved steatosis and fibrosis. However, there is no reduction of the energy expenditure and food intake. These results indicate different mechanisms existing for amelioration of MASH. Next, we examine the chronic kidney disease, CKD, using our collagen 4A5 R471X knock-in mouse model of CKD. This model carries a clinically relevant mutation, making it more translatable than traditional knockout mouse models. The phenotype includes albuminuria, elevated BUN, decreased GFR, and kidney failure. Irbesartan, angiotensin receptor blocker, significantly reduced albuminuria and its AUC, also suppressed pro-inflammatory and pro-fibrotic gene expressions. So here, these findings confirm the translational relevance of this model for CKD drug testing. In this section, I explain clinical diagnostic tools and parameters. In humans, modern diagnostic tools such as weighing scales can estimate, as you know, body fat percentage, muscle mass, and even bone density. To achieve comparable measurements in clinical research, we can use echo-MRI system, which enables invasive and non-anesthetic assessment of body fat and lean body mass in small animals. For more detailed body composition analysis, we also utilize X-ray CT. This approach, performed under anesthesia, allows us to distinguish between visceral and subcutaneous fat, as well as quantify bone and muscle mass. Importantly, both mouse and rats can be analyzed with these systems, providing highly translatable readouts that parallel clinical diagnostic evaluations. In the clinical evaluation of chronic kidney disease, glomerular filtration rate, GFR, is essential diagnostic and prognostic indicators. To achieve translational relevance, we apply the same principles in our preclinical models. Using plasma FITC levels sinistrin clearance, we can measure GFR in both wild-type mouse and disease model mouse, providing a direct functional readout of renal performance. As you can see figures, our collagen 4F5 mutant mouse showed significant reduction of GFR along with age. By adapting the same endpoints as those used in the clinical setting, our approach ensures that drug efficacy in preclinical studies can be more accurately linked to therapeutic outcomes in patients. In this section, I will introduce our comprehensive evaluation approaches on efficacy, safety, and MOA analysis. So nausea and vomiting are among the most common adverse effects of GLP-1 receptor agonists in humans. However, rodents don't vomit, which poses a major limitation for preclinical safety evaluation. To address this challenge, we established a comprehensive assay system that mimics multiple aspects of human emesis. This includes the conditioned test aversion test, CTA, gastric emptying assessment, calorie intake behavior, and salivary amylase secretion. Together, these assays allows us to capture both behavioral and physiological correlation of nausea in rodents. As you can see here, GLP-1 analogs demonstrated a response in these tests. In CTA test, they showed a response even at low doses, and also exhibit effects in gastric emptying test. They also increased calorie intake, a behavioral indicator of adverse stimuli, suggesting an unpleasant effect. The salivary amylase secretion assay is a unique assay system developed by our group. It measures a biomarkers or autonomic activation linked to nausea and vomiting. GLP-1 agonists stimulated amylase secretion even at low doses. This assay mimics multiple aspects of human emesis. To gain deeper mechanistic insights, we conduct double staining for brain neurons by in situ hybridization and C-force activation by immunohistochemistry. This approach identifies hypothalamic neuronal activation pathways relevant to appetite suppression. These mechanistic studies provide evidence of mode of action and enhance the translational value of our clinical models. To understand the mechanism of its anti-obesity effect, it is important to investigate whether it is based on reduced calorie intake due to suppressed appetite or whether it affects energy expenditure, oxygen consumption, and the utilization of fat and carbohydrates. We also apply indirect calorimetry systems named Oxymax systems to evaluate energy expenditure and respiratory quotient. Using diet-induced obese rat, we demonstrated beta-3 agonist that increases energy expenditure and reduced RQ. To evaluate energy expenditure is important analysis considering MOA for both obesity and MASH agents. Therefore, this analysis strengthens translational predictability in metabolic phenotyping. Finally, cardiovascular safety is a crucial part of translational pharmacology. We employ telemetry systems in rodent to monitor blood pressure and heart rate, as well as echocardiography systems for both small and large animals. These approaches ensure comprehensive evaluation of cardiovascular risk, allowing early detection of potential adverse effects. In conclusion, Axley's strategies emphasize three key points. First, highly translatable models reflecting human pathophysiology symptoms, disease phenotypes. Second, comprehensive approach links efficacy and safety evaluation with clinical outcomes. Third, broad applicability across pharmacology and diverse therapeutic areas in drug discovery. By adapting these approaches, we aim to maximize the human predictability of animal studies, accelerate drug discovery, and improve the chance of clinical success. That's all my presentation. Thank you very much for attention.
Thank you very much, Yasunori. All right, moving into our Q&A here for you. Our
Yeah,
first
of course.
question.
Mhm.
Perfect. Regarding the side effect evaluation of GLP-1 agents, I understand that the original amylase activity system developed by Axelid is used. Could you please provide more details?
Okay, I will. Thank you for question. It is important, and I will explain the detail of the uh, salivary amylase activity systems. This slide shows the detailed explanation of the rat salivary amylase activation for prediction of the emesis activity, as I mentioned. So, activation of the sympathetic neurons system, particularly through norepinephrine release from adrenal medulla, stimulates the parotid glands and enhances amylase secretion. We collect saliva by absorption into a cotton ball and measure amylase activity as a quantitative biomarker. Using this method, we can detect autonomic activation induced by nausea and vomiting. Related stimuli in rats using cisplatin and apomorphine which are famous MSS inducer in human. Importantly, this assay provides a non-invasive and translationally relevant approach, linking rodent response to human emetic mechanisms. Thank you for question.
Thank you, Yasunori. Another question here. Have you ever evaluated drugs other than ARBs in collagen 4A5 R471X mutant mice?
Yeah, that's a good question. Yeah, as I mentioned, the result of the ARB irbesartan, in addition to the ARB irbesartan, yeah, as you know, the dual antagonist of the endothelin and angiotensin receptors named sparsentan has been reported to be effective in collagen 4S3 knockout Alport syndrome mouse models. Although we can't show the data here today, yeah, we also confirmed the sparsentan's beneficial effect on kidney damage in our collagen 4F mutant mouse model. Furthermore, using our mouse model, Professor Nozu of Kobe University has reported the efficacy of exon skipping using antisense oligonucleotides. Therefore, we have confirmed the efficacy not only irbesartan, but also other drugs using this model. Thank you for question.
Thank you, Yasunori. And our final question for tonight. You measured the heart using an ultrasound diagnostic device. Have you ever used this device for other diseases?
Yeah, that's also a good question. Thank you. In addition to measuring cardiac function and morphology, pulmonary arterial pressure and right ventricular pressure are measured using echo in pulmonary hypertension models. Furthermore, in orthotopic ovarian cancer model, echo is also used to measure the size of ovarian cancers. To evaluate respiratory function in muscular diseases, the diaphragm movement is also measured by our ecosystem. Axelis has highly advanced ultrasound diagnostic equipment and experienced experts, enabling measurement of nearly all clinically evaluated parameters. Thank you for questions. I appreciate.
Thank you very much, Yasunori, for an excellent segment as well. And thank you also again for Manami for your segment. That is all the time we have today. If anyone submitted a question that wasn't addressed, keep in mind that the speakers will reach out to you directly. This session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. And before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. On behalf of EBD Group Bioeconomy, part of Informa Connect Life Sciences, thank you and have a great day.